This is just a distribution of the number of documented ecosystems that are hypoxic. And I think Vladimir really hit the nail on the head this morning when he suggests that it's probably due to investigator bias, or just where we've been looking for it. And in this paper, we also looked a bit at kind of authorship trends and kind of found at least some evidence that that's the case. So again, I'm really trying to, with my group, look at filling that gap in knowledge. And so today I'm going to touch on sort of three aspects, what I call the 3 R's of hypoxia and its impacts on coastal ecosystems. So that includes R for resistance, and I'll be focusing on corals in particular and how they respond to hypoxia. The resilience of coral reef ecosystems as a whole, looking at other components as well to low oxygen events, and then thinking about some of the ecological feedbacks that are responsible for driving patterns of recovery following hypoxic events. So just briefly, why temperate versus tropical ecosystems? We know that tropical ecosystems are important for a lot of the same socioeconomic reasons that temperate ecosystems are. They have a lot of similarities in terms of the contributions of various factors to driving the formation of hypoxia. There's a few differences. So in addition to them being relatively poorly studied, as I mentioned before, they tend to have consistently warm waters. And we do know that on a number of ecological and physiological levels, warmer waters exacerbate hypoxia. And we have a paper in Global Change Biology that really focuses on that, a paper I wrote with Karen Gadan. And they differ in terms of the relevant habitats of study. Okay, and so seagrass meadows are kind of common to both temperate and tropical systems, but there's also mangroves and coral reefs in the tropics. I'm going to focus on reefs for the system, but I am going to just showcase some of these other habitats as well, which we address in greater depth in some of the reviews that we published recently. So here's some pictures looking down from above on these various tropical habitats, and you see that there's these enclosed coastal ecosystems, which, as many of you know, is likely to promote hypoxia. And when you take a closer look in each of these, you see those same kind of diagnostic microbial mats that are signifying hypoxic stress in these systems. And so it's easy to find, and these hypoxic conditions tend to be widespread. Focusing in on coral reefs, at a variety of spatial scales, you see that there's tremendous range in oxygen concentration, but it can dip very low. So we termed this, we termed oxygen to be this universal currency on coral reefs. This is going to be true of other ecosystems as well. But that's because oxygen is constantly being produced and consumed, and is oftentimes a medium of transaction between organisms in the system, and it's oftentimes limiting. This figure is showing ranges in dissolved oxygen concentrations that have been recorded in reef systems at various spatial scales. And you can see that oftentimes becomes quite low. And as a consequence, you can see these very dramatic repercussions. At the top, it's showing how these healthy, vibrant corals, is from my study system in the Caribbean coast of Panama, went from these vibrant live corals to being bleached and then to being fully dead. And it wasn't just corals that were affected. There's these other reef creatures, such as these crabs and sponges, that suffered mortality as well. When you look globally at known hypoxic systems in the tropics, we know that there's coral reefs found in at least 50% of them, and that's just overlaying coral reef data sets with where there's known hypoxic systems. And the number of reefs that are likely to be affected is quite high. This is a, there we go. Just kind of a back of the envelope calculation based on kind of some large scale kind of geographic associations between reefs and coastal morphology. We estimate that globally about 13% of coral reefs are at risk of hypoxia. And that's easy to do for this particular number. We just went and we looked at kind of open ocean kind of reefs that are well flushed with open ocean water are at relatively low risk, and a reef that falls in an enclosed bay adjacent to a golf course is likely to be at high risk. And as I said, this was a very broad kind of estimate, but then Ariel Pesner, and I hope I'm not going to steal your thunder, but I saw her give a presentation recently at the Ocean Sciences meeting. She basically came up with the exact same value with by a completely different method. Okay, and that sent shivers down my spine and led me to think that there's some veracity to that number, and it's really kind of an estimate that we should go on. And there's severe consequences for reef ecosystems. So there's at least 26 instances where hypoxia has been implicated in mass mortality events. And there's these dramatic pictures. These come from the Indo-Pacific. It's the South Pacific, where we had high mortality of clams, some of which are being raised for in aquaculture, and then mass mortality events of fishes. Now, some of these events are sort of a consequence of natural processes. Others are certainly being exacerbated by eutrophication or organic material inputs. And in our review in coral reefs, we kind of like looked at the relative importance of those various drivers. But there's really a gradient in responses. So it could be a little bit more subtle than that. This is just a general cartoon, but it's just to emphasize that there are some sublethal effects and behavioral responses that shouldn't be a surprise to any of you based on the work that we know from temperate ecosystems, but certainly applies to coral reefs as well. And so it's in this subtlety and range of sublethal effects that we've decided to explore a little bit deeper. And I'm going back to this study system that where we've been working on the Caribbean coast of Panama. We find that there's a gradient in dissolved oxygen concentrations indicated by this color scheme, where the warm colors, the reds and oranges, are hypoxic, greens and blues are relatively high oxygen concentrations or normoxia. And what we see is that the species richness shows a strong relationship with the dissolved oxygen concentrations, where you have lower diversity at this end of the spectrum where oxygen levels were low during this particular event. But there still are some species that are able to persist. And so we've done some experiments to look at the tolerance to better understand species or interspecific variation in hypoxia tolerance. This is work that I did conducted in collaboration with Maggie Johnson, who led this portion of the study I'm going to talk about now, and Val Paul, based at the Smithsonian Marine Station in Fort Pierce, where we set up a mesocosm array where we have control of dissolved oxygen. We've also added some other factors as well that we'll be exploring in greater detail over the next year or two. But we looked at 2 different species that we thought might differ in their tolerance, and we did 4 different oxygen concentrations. And what we find is that there's a dramatic difference between these 2 species. So Cropper cervicornis, after just a few days, starts to show tissue loss. So within about 5 days, it gets to be about 50% of the coral tissues being lost, whereas Orbicella fovulata, relatively high tolerance. So after 11 days, there's no loss of tissue whatsoever. Here's a figure that just shows that tissue loss. But what you see is that it's only loss in the most severe oxygen treatment. So even if it's relatively depressed, let's say 2 milligrams per liter, you still have these both coral species persisting. There's not even tissue loss figure here for Orbicella because it is so tolerant. These trends extend to bleaching as well. So bleaching, which is typically associated with thermal stress, we find is also a consequence directly from hypoxia. And so what I would just suggest briefly is that oftentimes the bleaching episodes are easily or kind of assumed to be due to warming may actually be due in part, at least, to hypoxia. It's of hypothesis that needs to be explored in most systems. Most studies where bleaching has been documented. But we see is that there is bleaching, but only the most severe hypoxia treatment. And so there is some resistance or tolerance in both species, particularly in Orbicella. And so these corals are able to persist pretty well. There's been some other work that's come out of Sugget's lab, this paper by Alderis, that suggests that there's these hypoxia-inducible factors. And this is sort of their attempt to explain at the sort of cellular level what is controlling the susceptibility to bleaching. Okay, so we know that there's interspecific variation, that corals do have some resistance to hypoxia. But how do communities as a whole respond? And this has sort of been prompted by some work that was published by Dan Connolly and Bob Diaz that is showing that there's this expectation for hysteresis, or that the return trajectory of these communities may be different after being exposed to hypoxia than the initial impacts. They developed these generally based on temperate ecosystems. We suggested something similar could happen in coral reefs. And in this case, we suggest that coral reefs will have some subtle, not a sudden drop off in their the abundance of corals, because there's obviously some species that are relatively tolerant. So it'll be this kind of graded response. and that over time with colonial regrowth and lower recruitment, you can get a return back to the original state following an event. We had an opportunity to test the response or recovery, the resilience of these systems after an event that we've tracked in 2017. Just a picture showing the dead urchins and other reef creatures and bleached and dead coral during this event. We had 2 sites that we tracked across. This is essentially the same gradient that I described before, but this is years later. Collected data, and it was Noel that ran out and collected this with a YSI doing casts, and we tracked coral and the microbial community this time at the impacted site and at a reference site that was offshore where oxygen levels remained relatively high. At both these sites, we went out and quantified the benthic community. Okay, so we included everything, including say sponges, but with a focus on the corals in the system. We also looked at the microbial community. So we did sequence for 16S to look at overall microbial diversity, and also did some metagenomes to try to get a handle on the function of some of the genes that were present in the dominant microbial groups. At the reference site, I'll focus first on the benthic data. At the reference site, the benthic community was relatively unchanged through time. So we collected data before, during, and 1 year after this event. Going to the impacted site where oxygen levels dropped, we see that there's this shift. So before is in purple, during is red. 1 year later, the community is in that kind of teal color. And this is largely driven by the corals in the system. So you see that after a year, there's been some resilience, some return, but not full recovery in the system. Microbial community shows a very different response. This is where things get really interested. So first, just to get you oriented, these horizontal strips are individual samples that were taken at both the reference and the impacted site during the event. So these are water samples that were collected within 1 meter of the bottom. That were then filtered and sequenced. This is a 16S data. It's just showing each vertical bar is an amplicon sequence variant, ASV, essentially a taxonomic unit that was found there. And what you see is that the profiles for the communities are very different at the reference and the impacted site. You see that at the impacted site, most of the communities skewed here to the left, whereas over here you see a different set of microbes, different taxa that are relatively abundant. And this solid bars at the bottom are showing you which were relatively enriched compared to the other site. Okay, so you see there's these very distinct communities. Come back just 1 month after this event. And what you see is at the reference site, now those are the 3 samples are just pooled for simplicity here. You see that, not surprisingly, 1 month later, the microbial community looks very similar to the way it did during the event. Again, this is at the reference site. When you go to the impacted site, you see that the microbial community looks just like the reference site. So what it appears has happened here is that the microbial community has, within just 1 year, it's whiplash and it's gone right back to its kind of pre-impact community composition. We looked closer at some of the ASVs that were dominant at the impacted site during the event and did some metagenome assembly, and we find that there are genes for sulfur oxidative pathways. So it seems that there's some anaerobic bacteria that are responding favorably to these conditions, like opportunistically increasing in abundance or in presence at the system, in the system during hypoxia. So to summarize, using the same scheme that I showed previously, we find that these were our initial expectations, just generally for kind of coral reef ecosystems at the top. And we find that we found very different responses for the microbial and the coral communities. The coral communities impacted and boom, right back to where they started just a month later. A year later with the coral community, we're still on the way back. And what you see here is that even though normoxic conditions have returned, the community is not yet back to where it started up here. Okay, it's just starting its slow return. And so very different responses. And just point out here that what's clear is that the coral and the microbial communities are decoupled in this system. Given that microbes can play a very important role in the overall function of the reef, it's still yet to be determined what role they may be playing in the return of the system as a whole. But it's clear that they're decoupled, and it's worth investigating how these two community types are responding. All right, so what process govern recovery? Okay, so I already indicated that a year on, recovery is still taking place with the coral community at least. And I'm gonna just kind of take a step back and think more generally about reef ecology. something I think Denise alluded to earlier today in her talk. The expectation on coral reefs, just in general, is that when you have a large proportion of the corals die, that there's going to be a phase shift, and that the coral reef is going to transition to an algal community. Okay, so this is something that's been this paradigm that's been sort of dominating dialogue in the coral reef community for a number of decades now. And so what we hypothesized was that after hypoxia, there should be this phase shift into an algal forest. Okay, that the reef should look like a chia pet. And the question is, can grazers potentially stop that from happening? And the reason why grazers are thought to be so pivotal is that in coral reef ecosystems, the reason why this phase shift is thought to happen is that's a relatively new phenomenon over the recent decades because of overfishing of large herbivorous fish, such as parrotfish, and because of disease that killed diadema back in the 80s. And that has left algae to kind of free to kind of take over a reef. When the corals die, you got all that available real estate and the algae is going to move in. This is exactly why this current diadema epidemic that's sweeping through the Caribbean yet again right now is of such concern. And these are relatively large organisms. You have these large parrotfish that are targeted for food, and these large diadema that are, basically two that would fit in your hug. Okay, not that you'd want to hug one of those, but that just gives you a sense for how large they can be. So just to reiterate, the expectation is that after hypoxia, the reef should look like this. It should be this like kind of chia pet version of a reef where all the available dead coral substrates taken over by algae. But instead, what we found on these reef systems is that even after years following a hypoxic event, the system looks like this. I mean, clean as a whistle. Lots of available real estate. Why are there no algae there? We did a series of experiments. This is just the one that's sort of, I think, most photogenic. We transplanted 5 species of algae that you can find around the system if you know where to look. Did a little Easter egg hunt, found some algae, put 5 species out on the reef in one of these areas that had been affected previously by hypoxia and where the reef substrate was largely bare, and come back within just a few days, and it looks like this. What you're seeing here is that those are the coral fragments that the algae had been growing on, but they've been licked clean, and it's largely because of this herbivore that you see, these little reef urchins. This is a Echinometra viridis, very small urchin, about the size of a ping pong ball. And we did some size selective caging, and we found that even if you exclude any predators larger than those urchins, they still get herbivory of those of that algae. And so it really appears that it's the small herbivores in the system that are driving this response. Here's the data that here are the data that go with this figure. What you see is a full cage at the top, relatively steady abundance of algae over the course of the experimental period that ran for about 3 weeks. However, if you have either a cage control, which is essentially a partial cage, or a fully open treatment, you see that within just a few days that the algae is reduced down to nearly nothing. The exception is Halimeda, which is, if you're familiar with it, is this very pungent chemically defended species. It's also calcareous, so it's physically defended as well. And because it's so defended from herbivores, it was left alone in these experiments. So it's sort of this exception that actually proves the rule that herbivory was so dominant in the system. And so when you go and you census the herbivore community in Bocas del Toro, on the Caribbean coast of Panama, where we've been working, you see that the herbivore biomass is actually about 2 to 3 times higher than in other Caribbean ecosystems. And it's all these little herbivores. It's mostly the reef urchins, the Echinometra, the little ping pong ball urchin, striped parrotfish that get sexually mature at about the size of your finger. Okay, so these are relatively small diminutive species. Here's the data that goes along. This is first showing their average sort of body size, order of magnitude smaller than those large parrotfish that are harvested for food. And when you look at the overall herbivore community at these sites, it's really concentrated in these smallest size class or body size bins. So it's the same, the community is dominated by these really small herbivores, and they have the potential to keep the reef free of algae. This is some other work that I did. This was led by Hannah Nelson, where we went out and we looked at those reefs that were impacted by hypoxia. That's these sites on the right side. We also went to some healthy reefs that were on the other end of the gradient at this site. And we were able to compare live and dead coral areas as well as sand, so without any kind of complex habitat. And we compare that to the degraded sites that either had dead coral or sand, no live coral there to make that comparison. And this is just showing the relative abundance of reef invertebrates associated with these different habitat types. And what you see is that you get lots of invertebrates if you've got some structure there, whether it's live or dead coral. If there's sand, you get very few. This is looking to get across the broad taxonomic groups here that I've color-coded both in the photo and in the in the figure above. And this is important because that dead coral, after it's been impacted by hypoxia, is providing habitat for these invertebrates, including our friend, the little ping pong ball sized urchin over here. And so it is a large proportion of the abundance and the biomass of invertebrates in the system. Okay, so they're really taking advantage of this habitat, sheltering among the coral, and they're grazing the algae that's on the reef. Okay, so it's very abundant. It can number in the hundreds per meter squared in the system. Some other work that Noel has done followed up on this by testing their hypoxia tolerance and their behavioral responses or to hypoxia. Which he found is that it has the same or higher hypoxic tolerance as other urchins found in Panama, and that they're relatively good at maintaining their mobility and movement even when stressed with hypoxia. This is showing a riding response up here, that they're able to still like respond and move around as normal, even when stressed with hypoxia. Okay, so what I'm getting at here is that there's these feedbacks that have the potential to promote recovery after disturbances that include hypoxia. Okay, so open space, so you have coral, whether live or dead, is able to shelter fish and urchins. Those urchins maintain open space by grazing algae. That open space allows an opportunity for corals to recruit and to grow, and basically available substrate for them to attach. And so our ongoing research is looking at to what degree this is playing out. Are we actually seeing recovery in the system, given that this feedback appears to be in place? So we have to stay tuned. I'll come back in a couple of years with an update on that, because we're maintaining our long-term monitoring sites. And I'm not going to go into a lot of detail, but I just want to suggest that these feedbacks play a very important role in a number of these tropical ecosystems. And these are just several that we highlighted in a review. This is not all from our work. But in both seagrass meadows, coral reefs, and mangroves, is these positive feedbacks that promote recovery following hypoxic events that can afflict all three of these ecosystem types. But there's also some negative feedbacks. Those are the feedbacks that maintain kind of a degraded or hypoxic condition in these systems. And that's why you end up with, to some degree, some form of this hysteresis, where once you get the transition, you have that hump you have to overcome, while you're a little less likely to get a return in some systems than others. And it depends on the nature of these feedbacks and their longevity in whether you get these transitions. And so what I'm trying to get across, both in this review that we published as well as right now in this talk, is that we need to think about the entire community, not just the habitat dominance, but maybe microbes and other players such as the herbivores, and think about it in the context of these feedbacks are likely to promote either a degraded state or promote persistence in a healthy state and help with the transition and recovery back into the system. Okay, so just quick summary here. So conclusions for the 3 R's in coral reef deoxygenation. Coral reefs are vulnerable to deoxygenation. We've seen that there can be these mass mortality events, but there's also some resistance. There's some hope for optimism because some species are able to resist low oxygen stress, particularly if it's only intermediately hypoxic. Hypoxia can result in community state shifts for both corals and microbes, and likely for other components of the community as well. But these components show a decoupling or differences in their potential for resilience, and so their recovery is likely to be at different paces. And there's pathways and ecological mechanisms of recovery that are likely to differ from the initial impacts of deoxygenation. And what I mean by that is that the transition from a coral state to this bare coral, or even the potential for an algal state, is being driven largely by a lack of dissolved oxygen. But the recovery is going to be dependent on more than just the return of oxygen to the system, but you need these feedbacks involving the entire community and its interactions to allow for recovery and to predict the pace. So final slide here, just some of these same points along with future directions I suggest are needed. These are not mutually exclusive, nor is it comprehensive, but based on our research, it's 3 directions that we see the need for further research. We need to better understand physiological mechanisms interspecific variation. We've done a pretty good job of developing that in temperate ecosystems, although there's plenty of work to do, as we just saw in the previous talk with the dogfish. But particularly in tropical systems, we need a better handle on that to make some predictions and which species we might want to focus on for management and conservation priorities. We need to improve knowledge of microbial functions and timelines, and so how that plays into timelines of community recovery. And we also just more generally need an integrative perspective that considers not just the physiological response of organisms, but also ecological feedbacks, and really looking across different levels of ecological organization from the organism up to the ecosystem. So with that, if there's time, I'm happy to take any questions, and thank you for your attention today.
Thank you so much for a really wonderful talk. Work like this has really improved our understanding of low oxygen issues in coral reefs. We do have time for a question.
Thank you very much for, can I go?
Yeah.
Thanks a lot for the great presentations. And I have a questions about the model that you present in the hysteresis graph. So you presented the anoxia, the hypoxia, and the normoxia. Which is the role of hyperoxia that occurs in coral reef to drive the dynamics of community?
So you're asking what's the level of hypoxia that can be experienced?
No, which is what do you think is the role of hyperoxia that coral reef?
Oh, hyperoxia? Yeah.
Hyperoxia, supersaturations.
Thank you. Right. So the question about hyperoxia, which is supersaturation of the seawater. In the review paper that we published in Coral Reefs that was led by Hannah Nelson, she really focused in on hyperoxia quite a bit. And a lot of relationships between oxygen and function of corals is essentially a unimodal relationship. And so there are certain factors like calcification and productivity that will decline as oxygen levels drop. There's some optimum, and they actually decline as oxygen levels get too high, because you can get over 200% saturation, for example, on the reefs, and that can be detrimental. And so there's different physiological mechanisms for trying to deal with that stress in corals. And yeah, so it is a stress, and it is prevalent on reefs, and as it is in a lot of high productive systems in temperate regions too. Thank you.
Very quick.
Yeah, excellent. Thanks for your talk. And for warming, or we have used the warming or heating days metric to have some accumulation of stress. Do you expect that for oxygen being the case as well, or is it instantaneous? And what do you think drives really the whitening or the bleaching of the corals?
Yeah, so that's a good question. I think it comes down to an issue of recovery. And so whether it's just the cumulative number of days that are hypoxic over say a summer season, for example, or a wet season. Yeah, I think it depends on the recovery of the species. That's something I don't have a good handle on. I don't know how well that's understood by others, but in a reef system, I think it's going to depend on the interval between events. And so in temperate systems where I've worked, such as Narragansett Bay and on the east coast of the US, we saw that hypoxia tended to occur during when there's minimal tidal amplitude. So you have like a week or two of stress and then a week or two of of a break. And the reef systems we're working, we're still getting a handle on the the duration of those events. They can last a few days, but they can potentially last longer. And if the events are recurrent, then it's going to depend a bit on the recovery and the length of that interval. And so it's going to be important. It's not just the total number of days, but I don't have a good handle on numbers for you yet. Yeah, thank you. It's a good question.
Thank you so much. Yeah. We need to move on to our next speaker, who is remote. Noelle Lucy, who is from Smithsonian Tropical Research Institute, where Andrew used to be. He's now at University of Florida.
Hi, everybody. My name is Noelle Lucy, and I'm coming today. Hi, everybody. My name is Noelle Lucy, and I'm coming today to talk about oxygen extremes. Environmental extremes, as we know, long-term habitat structure is the result of an interaction between temperature and oxygen. So with this research here, I'm presenting you with a mechanism that explains how short-term ocean extremes are impacting species biogeography, particularly on shallow tropical coastal coral reef habitats. And I'm working in Panama. I'm on the Caribbean coast of Panama, close to Costa Rica, at the Bocas Research Station with the Smithsonian. And this overlooks a large bay, Almirante Bay, which you can see here in this map. This is another map of Almirante Bay with the distribution of two brittle stars overlaid. So you see in yellow Ophioderma cinerarium and in blue Wentii. And what we have is in the inner bay reef sites at 3 meters depth, the reefs are dominated with cinerarium in comparison to Wentii. And as you get closer to the open ocean, we find more Wentii occurring. Why is this? They have very similar ecological traits, fill ecological niches. So perhaps it's temperature or oxygen or some combination of the two that's responsible for Wentii's exclusion from the inner bay. In order to determine this, I looked at both species' upper thermal limits. So their non-lethal upper thermal limit as their CT max. And I did this under hypoxia and normoxia. So we just ramped up the temperature, waited for the arms to curl up and become unresponsive. At that point, we deemed this the CT max and found that Wentii actually had a much higher thermal limit than cinerarium. So that's the blue line under normoxia compared to the orange line here. And in hypoxic conditions, the two species had very similar thermal limits converged, which doesn't tell us that temperature is responsible for Wentii's exclusion from the inner bay. So it could be oxygen or hypoxia sensitivity. Wentii might be too hypoxic sensitive, and that's why it's not in the inner bay. In order to test that, you would think that we could find the P-crit as a proxy for hypoxia tolerance and see differences there. So we used closed chamber respirometry to do this with both species at ambient temperature and under high temperature. So these pulses at different temperatures were just done for 4 hours. And we found that the P-crit for both species was the same under ambient temperatures, and counterintuitively, under higher temperatures, Wentii's P-crit actually went down, indicating that it's more hypoxia tolerant than cinerarium, which doesn't really explain why it's not in the inner bay. These results don't really show a very clear picture of Wentii's true hypoxia sensitivity, though. We did behavioral and survival assays as well and found that if you put both species one after another into hypoxic chambers or water chambers, and then pumped in deoxygenated water to create this gradient where you have hypoxic water at the bottom and normoxic water at the top, we looked to see if the animal could sense and then crawl up this ladder to escape this hypoxic environment at the bottom here. And we found that when Wentii mostly hung out at the bottom, cinerarium was 2.5 times more likely to sense and escape hypoxia. You might think Wentii is hypoxic tolerant, so it doesn't need to escape. However, we did survival assays where we subjected individuals to hypoxia for 4 hours and found that almost every single Wentii individual died, whereas every single cinerarium individual lived, which clearly shows that Wentii is not hypoxia tolerant. So we can't use these traits to explain Wentii's exclusion from the inner bay, but we know that thermal limits and hypoxia tolerance are key in predicting species' geographical ranges. So we argue that there's another trait based on the metabolism of a species that might be more accurate at predicting species whereabouts based on these short-lived extremes. And this is the metabolic index. which is simply just the ratio of environmental oxygen supply to organismal oxygen demand. And it's calculated using the temperature-dependent hypoxia tolerance of the individual or the species, which are shown here in the blue boxes of this equation, as well as the temperature and oxygen that is in the environment that the species is in. So this equation gives you phi, which is just a factor by which the oxygen supply can sustain the metabolic rate of a given species above either a resting or active state in a given environment at a given time. So in order to determine both species' oxygen requirements for activity and use the metabolic index to look at this, we physically exerted both species, and then we measured how much oxygen they needed when they were active. And we found that Wentii actually need 2.5 times more oxygen than Cinerium under anormoxic conditions. So see the blue line in contrast to the orange line here under normoxic conditions. And under hypoxia, the aerobic scope of Wentii was significantly depressed. So this decrease in aerobic scope with strenuous activity under stressful conditions really does indicate that Wentii needs far more environmental oxygen to fully perform its ecologically important activities such as reproducing, getting food, evading predation, you name it. So with these traits, we were able to determine when the environment isn't meeting each species' energetic demands. So we measured this as the hypoxia exposure. So the environments we focused on are shown here in red and blue, the inner bay site and the outer bay site. So the extreme ends of the oxythermal gradient of the bay. And we have phi calculated at these two sites over diel and annual timescales. So diel on the left and annual timescales on the right. And you have the factorial aerobic scope for the species as the dashed black line at the bottom of these plots. When phi, or the metabolic index calculated for this species at both sites, falls below the factorial aerobic scope or the dashed black line, this indicates the activities of this species at that temperature would be limited by local oxygen availability. And for Cinerium, hypoxic exposure in both sites was very low. So it didn't, the occurrences of low oxygen didn't occur very often in either the inner bay or the outer bay. However, here we have Wentii using the same time series, and we find that its hypoxic exposure is significantly higher in the inner bay during both time scales compared to Cinerium. So that at night, it's limited 12% of the time. So you see these values under the line. And during the day, it's also limited 10% of the time. It's usually in January and February and October here. So this shows that the inner bay reef is metabolically restricting the activity of Wentii. We wanted to link these occurrences of hypoxic exposure to climate. So we needed to look more closely at the relationship between oxygen and temperature. So we quantified oxygen variability and temperature by mapping out the total number of times each oxygen and temperature measurement combination occurred during the last decade. You can see this here simply as with the red indicating average or frequent occurrences and the light yellow or white as rare occurrences. And this really clear shape emerges, the wedge. It's where you have increasing temperature, both positively and significantly correlated to increasing oxygen variance. Okay, so back to our species. This is the wedge again. Now you can focus on the exclusion thresholds that we've drawn for each species on this reef environment. So the blue line here is Wentii's exclusion threshold, and the cyan color is Cinerium's exclusion threshold. And what I mean by exclusion threshold is that it's the threshold by which each species can't, any conditions that are under its exclusion threshold limit the activity of the specific species. So here you have the current exposure of Wentii being 8 times greater in the inner bay compared to Cinerium, with all of these occurrences happening under the blue line compared to the cyan line. So we modeled this relation between temperature and oxygen variance. You see the fit on the top right plot. And we use this to project what would happen if temperatures warmed by 1 degree. And we see that on the bottom left. And we find that the frequency and the severity of extreme oxygen conditions increase. So you have the values under both species exclusion thresholds increasing, and this is showing that as the wedge is moving to warmer temperatures and lower oxygen values. Here you have an increase in temperature of 3 degrees, and this increases the hypoxic exposure, limiting the activity of both species regularly. So all of these values are falling below both species exclusion thresholds, and this dramatically reduces the habitability for Cinerarium. So both species under warming of 3 degrees are excluded from the bay due to this increase in oxygen variance. So we know that warming is affecting these two species. This color field shows this again. It looks at the hypoxia exposure on the inner bay reef during the past decade. So you have the red colors indicating that most of the time it's oxygen limiting in the inner bay, and the white colors indicate that it's not. There's plenty of oxygen available. We've just calculated or shown overlaid the active hypoxia tolerance and temperature sensitivity traits onto this environmental space. And we see the species physiological traits for the two brittle stars are in the asterisks here, are shown as the asterisks here. And you have this black line as the current exclusion threshold based on a 5% hypoxia exposure occurring. So you have Cinerarium falling to the left of this exclusion threshold, this black line, and Wentii just to the right. So Cinerarium is still able to be in this environment, whereas Wentii is just barely excluded in this case. But what's striking is that if you increase the temperature and project a 3 degree warming with oxygen variance, you're going to move or shift this exclusion threshold from the black line to the red line, and that's going to exclude Cinerarium from this environment. Only, however, if you include oxygen variance in the projected temperature increase. If you don't, you see what happens here is this red dashed line occurs, and it does not exclude Cinerarium. So you're underrepresenting the habitability of the site for Cinerarium without by not accounting for the oxygen variance. Another striking thing here, and probably more importantly, is that all of the other species across the globe that have these physiological traits are shown as these other black dots in this trait space. And it seems like most of these species are unable to tolerate the oxygen conditions on this reef, as the majority of them are falling to the right of this current exclusion threshold. So you see them in the dark orange region of the plot. A couple of them are not. They're out here and seem to be okay, but would be excluded with future warming, accounting for oxygen variance. Caveat here is that most of these species that we have traits for aren't tropical, which just highlights how little we know about the tropical species that we have and don't have any idea really about their hypoxia traits. And that's what some of my new research is looking at. I'm trying to understand temperature-dependent hypoxia tolerance in reef-building coral species. These are just some preliminary results in different Caribbean corals, and their oxygen consumption rates as a function of temperature. And as you can see, as temperatures go up, they're consuming more and more oxygen, and this is happening at night when the animals are respiring. So as oxygen becomes more limited at night, and temperatures become higher, the corals are going to need more oxygen. So it begs the question, how tolerant are these species to hypoxic extremes? So we measured this with one species, Siderastria siderea, which is a hypoxia tolerant species, and mapped its exclusion threshold onto this inner bay environment. And this is the green line. You can see it with these different projections. And in a future context, we have a lot of similarities between the coral and the hypoxia tolerant brittle star, where under future warming, the hypoxia tolerant coral is also going to be metabolically challenged by oxygen limitations on this reef. So, in conclusion, we see oxygen variance significantly increasing with warming. It's causing oxygen extremes that are restricting the activity of coral reef species and causing aerobic range shifts. And our data really does, I think, show very clearly that if we ignore oxygen extremes, we're likely going to underestimate a powerful mechanism contributing to local extirpations on tropical coral reefs. With that, I'd like to thank you and all of my collaborators that made this work possible, particularly Curtis Deutsch, who has been working closely with me on this, and take any questions.
Thank you so much. I'm afraid that we're going to need to move on to the next speaker, but you can ask questions on the Padlet as well. Our next speaker is also remote, Brad Seibel from University of South Florida.
Right. Hello, everybody. Thanks for tuning in, or I wish I were present there with you, but just wanted to give a quick talk about hypoxia and hypoxia tolerance and how we define and measure them. This quote from Mark Twain, "It ain't what you don't know that gets you in trouble, it's what you know for sure that just ain't so." I feel like there are a lot of things that we have thought historically that we knew for sure that are turning out not to be quite so, at least. And so I want to focus on what I think are common misperceptions, and in particular one regarding the temperature effects of the effect of temperature on hypoxia and hypoxia tolerance. Let's see if they switch for me. Don't seem to have control just yet. Oh, there we go. So in the vast majority of papers on ocean deoxygenation, you'll see a figure something like this, or a statement something like this, that as temperatures warm, oceanic oxygen supply is diminished and hypoxia tolerance tends to decrease as oxygen demand increases. And this is true to an extent, but it depends on how we define hypoxia and hypoxia tolerance. So warm water holds less oxygen. We hear this all the time, but what is less appreciated is that a given concentration of oxygen exerts greater pressure at warm temperatures. So for example, in a parcel of water that's saturated at the surface in equilibrium with the atmosphere, the partial pressure of oxygen, which is the vertical axis here, as a function of temperature, the partial pressure of oxygen is constant at air saturation, or 21 kilopascals, across that temperature range. So at warm temperatures, that air saturated water holds only 6 milligrams per liter. That's these dashed lines or isolines for oxygen concentrations in milligrams. And at colder temperatures, that parcel of water would hold 10 milligrams per liter. Hypoxia is often defined as a lower threshold of near 2 milligrams. It varies depending on paper, but as you can see, 2 milligrams per liter exerts higher pressure at warm temperatures. So a given concentration is not equal across those temperatures. If you were to take that parcel of water at warm temperatures, remove it from equilibrium with the atmosphere and cool it down, it would exert, that same concentration of oxygen would exert a much lower pressure at lower temperatures. So this graph shows oxygen partial pressure as a function of month across the year for a representative temperature profile in an estuary. And you can see this curved line is the partial pressure exerted by 2 milligrams per liter across that monthly temperature variation. So in the summer months, 2 milligrams per liter exerts nearly 6 kilopascals of pressure. And in the winter months, 2 milligrams exerts only about 4. The dashed horizontal line represents the mean critical oxygen pressure for animals in that environment, which, as you'll see shortly, does not change with temperature. So in winter months, 2 milligrams per liter would actually be better for the average species there than it would during the winter months. Sorry, in the summer months, it would be better. They would be able to maintain some aerobic scope, meaning that the oxygen available is greater than their pressure required at rest by some factor. So that's the factorial aerobic scope. Whereas in the winter months, the oxygen pressure is lower than their critical pressure, meaning they would experience some oxygen debt. So the other side of that coin is that oxygen demand increases in warm water, which is also true. But what's less appreciated is that the ability to supply oxygen physiologically also increases with temperature. So this schematic represents the relationship between some of these metabolic traits we've been hearing about. So the standard metabolic rate is in blue here. And as oxygen declines, most organisms can maintain this standard or resting metabolic rate down to some critical low oxygen level, at which point it falls off. And death is usually not far behind at that point. So if you divide the metabolic rate by its critical pressure, you get the slope of this line, which is the oxygen supply capacity. That's how much oxygen they can supply per unit available. So if you multiply that slope, alpha here, by oxygen pressure, it describes the oxygen dependency of maximum metabolic rate. In other words, at a given oxygen partial pressure, this line describes the maximum rate that can be achieved at that pressure. So the maximum rate is measured usually under exercise or forced or in a swim tunnel. And there's a critical pressure for that maximum rate as well that we call P-crit max. And for most species that have been measured, it's somewhere near air saturation, which means that hyperoxia doesn't typically have an effect, although there are exceptions. So as oxygen declines to air saturation, the maximum metabolic rate is constant until it reaches that PC max. Below that, it follows this line. So if you increase the temperature, which follows this line labeled number 1, the maximum metabolic rate increases, and the slope of this line, the oxygen supply capacity, increases to meet it. So importantly, and as we just heard a few minutes ago, the aerobic scope, factorial aerobic scope, is usually measured as the factorial difference between the maximum and resting metabolic rates, but it can also be measured as the ratio of the critical oxygen partial pressures for those rates. So as temperature increases, the critical oxygen pressure for the maximum metabolic rate stays the same near air saturation, and oxygen supply increases to meet it. So this is the relationship between maximum metabolic rate and that oxygen supply capacity measured as the standard metabolic rate divided by the P-crit for that rate in about 50 different species. I've divided them by temperature, so you can see animals that were measured below 20 degrees tend to have lower maximum metabolic rates on average. Species that were measured at above 20 degrees have higher on average, but they all fall along this line. So if you are a more active animal, an oceanic squid or a tuna fish, you have a very high maximum metabolic rate and a high oxygen supply capacity to meet that. If you're a sluggish animal, a flatfish, for example, you would have a low metabolic rate and a low oxygen supply capacity. So increases in lifestyle mobility or temperature or body size per gram would have the opposite effect. A large animal would be found toward the lower end of this curve, and a small animal would be found toward the upper end of the curve. But oxygen supply increases evolutionarily or through acclimation to meet the maximum demand at whatever temperature or body size the animal is found at. So just as one example of what oxygen supply capacity consists of, these are animals that live in low oxygen environments that tend to have very large, high surface area gills. They also tend to have high ventilatory capacities and high heart rates. This is a video that I can't show you, but it's a gill of a squid that's a very active animal and also has high gill surface area to meet that increased demand. So that high oxygen supply capacity increasing with temperature effectively gives animals the same critical oxygen pressure for maximum metabolic rate regardless of temperature. So animals living at warm temperature have a peak crit max near 21. Animals living at low temperature have a peak crit max near 21. So regardless of how much oxygen concentration is held at those temperatures, the animals have evolved the capacity to extract the oxygen they need to meet their maximum metabolic rate. If oxygen declines, the maximum metabolic rate will decline in proportion. You can see critical oxygen partial pressure for the resting rate is also independent of temperature across species. So 2 milligrams per liter on average is sufficient to meet the resting needs of the majority of these species, where at lower temperatures it is not. So the peak crit max is the oxygen pressure below which oxygen begins to limit metabolism and aerobic scope. So the aerobic scope, the factorial aerobic scope, can be defined, as I said, as the ratio of the critical pressure for maximum to the resting. And if oxygen is reduced from 8 to 6 milligrams per liter at, say, 15 degrees Celsius, so that's a 20% drop, you get a 20% reduction in maximum metabolic rate and aerobic scope. If oxygen is reduced to 3 milligrams per liter at warm temperatures, these animals would still on average maintain aerobic scope of about 1.5, maybe up to 2, whereas at 5 degrees, they would have 0 aerobic scope. An aerobic scope, of course, represents the potential energy available, the potential to extract enough oxygen to support all of the needs beyond basic maintenance and rest, such as growth, reproduction, foraging. Now, acutely, the critical oxygen pressure does change with temperature. So each of these data points on here represents a different species. But if you were to look at any one of those species across an acute temperature range, the critical oxygen partial pressure increases, indicating a reduction in aerobic scope. So at a warmer temperature, aerobic scope is going to be less, and at a lower temperature, aerobic scope is going to be greater. But that's an acute response, and most organisms do not experience this sort of 10-degree Celsius temperature change acutely. So how this is interpreted in a climate change context is a little bit tricky. We don't yet know for most species what the capacity is to increase oxygen supply beyond the range they normally experience. So these are just some examples showing that same thing, that within a species, for example, Atlantic cod, the critical oxygen partial pressure at rest increases with temperature. For black sea bass, it's shifted to the right, but you get about the same range of critical oxygen pressures. So black sea bass living at warmer temperatures has a higher oxygen supply capacity to meet its higher metabolic demand, which protects aerobic scope. So it maintains the same level of aerobic scope as the Atlantic cod, but at a higher temperature with higher metabolic rates. These are estuarine species that tend to have slightly lower critical oxygen pressures, but the same sort of range and same and definitely overlapping. And we'll get more to estuarine species in just a minute. So in estuaries, oxygen varies quite dramatically on hourly, daily, monthly, and interannual sorts of timescales. And so you can look at the minimum oxygen value that's recorded across a year. You can look at the mean oxygen value recorded across a year, or the maximum, which tends to be a little bit supersaturated. And these are the critical oxygen partial pressures, which I'll come back to in just a minute. The maximum and the resting critical oxygen partial pressures for estuarine species. So you can see the minimum value that you experience throughout the year tends to drop below that critical resting level, but the mean tends to be well above P-crit max.
Brad, can we wrap up, please?
Yep, yes, we can. All right, so this is just showing oxygen supply capacity is higher in estuarine species, which gives them a lower P-crit max, meaning that that these animals do not begin to experience negative effects of deoxygenation until nearly half saturation. And their growth rates are similarly unaffected until you reach below half saturation. So I will stop there. Thank you.
Thank you so much. And again, you can ask questions of Brad through the Padlet. We have another remote talk, Emma Pontes from University of Miami, Rosenstiel School of Marine and Atmospheric Sciences, talking about critical partial pressure determination of corals.
Thank you so much. I'll just wait for my presentation to get uploaded. Okay. All right, so thank you so much for taking the time to hear my talk today. My name is Emma Pons. I'm a third-year PhD student at the University of Miami's Rosenstiel School, and I'm going to be talking today about the critical oxygen partial pressure determination of the scleractinian coral Acropora cervicornis. And I'm sorry I can't be there in person today, but I do want to say it's a pleasure and an honor to hear from the scientists that inspired this research and informed it as well. Let's see if I have control. Okay, so I'm sure we're all familiar with this idea that climate change is causing significant changes to the chemistry of our ocean. So human release of greenhouse gases have caused a significant change in the pH. We're experiencing ocean acidification. It's also caused a increase in the temperature of the water, 0.8 degrees Celsius since about the 1900s. And what I'm going to focus on today is the fact that we've lost a good amount of oxygen globally. So dissolved oxygen globally has gone down significantly. And I know this figure was shown by Dr. Altieri, but I think it's a really nice figure to just show, not necessarily very nice, but this idea that global loss of oxygen is causing coastal hypoxic events. And these coastal hypoxic events can become really severe. So everywhere where there's a red dot is essentially a documented dead zone where essentially the water got to 0 milligrams per liter, roughly 0, potentially maybe 1 milligram per liter, essentially anoxia. And these yellow dots represent where hypoxia was implicated in coral reef mortality. And so the important thing here is that these events are happening coastally, which is where coral reefs exist. So we know that these hypoxic events are going to become a really severe threat to coral reefs. And we've seen that happen. So I'm located here in Miami in South Florida. And we've seen a lot of hypoxic events causing mass mortality in this area. So it happened in the Gulf of Mexico and Flower Garden Banks. You can see from these photos some really kind of gnarly dead coral tissue that was caused by low dissolved oxygen arising from the benthos. As Dr. Altieri mentioned, it also occurred in Panama, where this really beautiful colony of Agaricea lamarcki over the course of a couple of days was succumbed to mortality. And then right in my backyard here in Miami, in Biscayne Bay, we've experienced these low DO events that have caused fish kill events. So these really low amounts of oxygen in the water is causing fish to basically die and float to the surface. So again, as Dr. Altieri mentioned, I'm including this slide because I wanted to point out that while organisms undoubtedly become stressed due to hypoxia, we know that corals have differential susceptibility to hypoxia. Some are more tolerant than others. And Dr. Altieri did a really nice job of explaining this study earlier. And I just wanted to point out that Acropora cervicornis is known to be relatively sensitive to hypoxia. So after only 5 days of exposure to severe hypoxia, that A. cervicornis was experiencing tissue loss, whereas the Orbicilla favulata, which is known to be a little more hypoxia tolerant, experienced no tissue loss after 11 days. So this was a really interesting study that helped kind of inform the research that I wanted to do. So again, I wanted to focus on Acropora cervicornis, mostly because of its importance here in South Florida. So Acropora cervicornis was the predominant reef building species in South Florida before the 1980s. And since then, we've seen about a 97% decline in their population due to things mostly due to disease, but also due to things like ocean warming, ocean acidification, and more recently, ocean deoxygenation. Another reason to focus on Acropora cervicornis is the fact that it's the main focus of reef restoration efforts. So if we can identify potentially different genotypes of cervicornis that are more tolerant to hypoxia than others, we may want to prioritize outplanting those genotypes on the reef. So all of this led me to ask the question, essentially, what is the hypoxia tolerance of Acropora cervicornis? And one of the ways that I wanted to go about measuring that was by calculating the critical oxygen partial pressure, or PO2 crit, which we've heard a lot about today. It's widely regarded as a measure of hypoxia tolerance, but it's more accurately described as a reflection of the organism's physiological oxygen supply capacity. So how well is that organism able to transport oxygen throughout its body to the tissues that actually need it? So we can define PO2 crit in a couple different ways. One of them would be where the metabolic rate becomes oxygen dependent. So essentially, and again, we've heard a lot about this in different talks today, but it's the point at which an organism can no longer maintain its normal rate of metabolism such that it must conform its rate to the environmental oxygen availability. This is also the point at which the oxygen supply capacity is at its maximum. It's working its hardest to supply oxygen to those tissues under oxygen limitation. Sorry, there's just a slight delay with my slides. So in terms of methodology, we use a closed system intermittent flow respirometry system. What we did was we defined 14 distinct oxygen levels. We attempted to reach whole numbers and 0.5 numbers in terms of milligrams per liter. And we essentially did the best we could with our bubbling system. So on the left over here, I'm showing this table that has our different oxygen values in milligrams per liter and also in partial pressure, as well as the sample size. So this indicates the number of corals that we had per treatment. And it's important to note that these treatments were run individually on a single day. So no two treatments were run on the same day, and corals were allowed to recover for 24 hours in between these treatments. So we expose the coral to the given treatment for about an hour. Then we use our closed system intermittent flow respirometry to measure the photosynthesis and the respiration inside those closed chambers. Photosynthesis was achieved just by allowing the coral to be exposed to light, whereas respiration we measured in the dark by covering the chamber with an opaque covering, so no light was being measured. Or no light was entering that chamber. So it's important to note here that with the photosynthesis measurements, we're really only looking at the oxygen expulsion of the algal symbionts that live within the coral tissue. Whereas when we look at the dark respiration, we're actually looking at the metabolic rate of the coral host. So just to give you an idea of what our setup looked like, we used a Vernier DO optical probe to actually measure the amount of oxygen in the water. We did have a bubbler that was attached to a nitrogen cylinder. So that bubbler was bubbling nitrogen into the water whenever the water got to above the level that we wanted it to be for that trial. I'm also including my couple little coral subjects in the bottom left corner, and then just a better photo of that respirometry chamber that's actually attached to a pre-sense sensor. So essentially inside that chamber, there is a pre-sense sensor that's measuring the amount of oxygen inside that chamber every 5 seconds. And it works together with a digital control unit and a solenoid valve. Essentially, I can use this Vernier LabQuest interface, which is basically a mini computer that lets me set the level of oxygen I want in the water. It then communicates with the digital control unit, which is attached to the probe. So anytime the probe is reading a value above the level of oxygen that we were trying to achieve, that solenoid valve kicks on, nitrogen gas begins to flow, and that's how we're able to actually lower the oxygen inside of our treatment tank. So in terms of results, what I'm showing here on the x-axis, we have the partial pressure of oxygen. So this represents the different oxygen trials that we ran. And then on the y-axis, I'm showing the metabolic rate. So this is just the dark respiration data that we're looking at. This is just the oxygen consumption from the coral host. And each one of these dots represents the average of all the corals in that treatment. We actually didn't see too much genotypic variability within our different genotypes. So we just wound up averaging all those corals together, especially given our small sample size. So what we found is we actually used the respirometry package in R to calculate our p-crit. And what that does is it uses the broken stick method, where it fits a line to the maximum metabolic rate, and it also fits that alpha line, which represents where the organism is conforming its metabolic rate. And so the intersection point of those two lines represents our PO2 crit, which we calculated for Acroporis cervicornis to be 6.9 kPa, which is roughly 2.5 milligrams per liter. I personally like to think about things in terms of milligrams per liter, which is why I included both values there. And so this is really interesting because, well, this is the first time that PO2 crit has been calculated for Acroporis cervicornis. And what we found is that it's actually higher than the literature definition of hypoxia, which is generally 2 milligrams per liter, which winds up being 5.2 kPa depending on the temperature. And again, this is concerning because this means that corals will experience metabolic suppression before a hypoxic event is actually recognized by the literature or by scientists that are kind of looking for that 2 milligram per liter value. So this suggests that this species is very sensitive. Again, this is kind of what the Johnson 2020 paper found, is that cervicornis is relatively hypoxia sensitive. I also wanted to do this calculation on the photosynthesis data. So this is the same graph. Here we're looking at the partial pressure of oxygen on the x-axis with the now photosynthetic rate. So here we're looking at the oxygen expulsion. I forgot to mention this in the previous slide, but we did fit a hyperbolic tangent function to the data in order to do these predictions. So again, we used the respirometry package in R to do that broken stick method that finds the intersection of these 2 points. And what we found is that the p-crit for photosynthesis was higher than the p-crit for respiration. So we calculated it to be 10.1 kPa. This is again a little concerning. This indicates that not only is the coral host susceptible and negatively affected by oxygen limitation, but so too are the algal symbionts. And those algal symbionts are potentially more sensitive than the coral host. So again, this this was an important calculation for us to do, but is definitely a little bit concerning. So in conclusion, we found that the p-crit for cervicornis was calculated to be roughly 6.9, which again is concerning because it's above that literature definition of hypoxia. So I think this points to the idea that one, corals will experience metabolic suppression before a hypoxic event is recognized, but it also highlights this idea that maybe that 2 milligrams per liter as a hard and fast definition of hypoxia is not necessarily a universal a threshold that we should be looking towards, and that there might be kind of species specific or genotype specific thresholds as well. And we also found that those algal symbionts are sensitive to changes in oxygen availability and potentially more sensitive than the coral hosts that they reside in. So in terms of next steps, before publishing this data, I plan to calculate the p-crit for several other species, including Psittaciaria psittarea, which we heard earlier is known to be relatively hypoxia tolerant, Orbicella faviolata, which we also know to be relatively hypoxia tolerant, Psittaciaria radians, Preides astroides, and Preides preides. And we chose the other Preides because we wanted to look at how different growth forms, different morphologies might affect the p-crit. We suspect that maybe branching species or more erect growth forms might be a little more sensitive to hypoxia than these flatter growth forms. So keep an eye out for that as we conduct those experiments. And I just want to thank my lab, particularly my advisor, Dr. Chris Langdon, my committee members, Dr. Andrew Baker, Dr. Diego Learman, and Dr. Ian Enox. A couple other faculty have helped me out, Martin and Danielle with the respirometry. And I really want to thank the Global Oxygen Network for allowing me to speak today. And with that, I'll be happy to take any questions.
Thanks so much. We've got time for some questions. Any of the remote people? Nope. That's
Yeah, thanks a lot. That was a great talk. I'm curious, do you have an explanation as to why photosynthesis was so oxygen sensitive as it was?
Yeah, that's a great question. And my advisor and I have been kind of trying to figure that out. And I think there's a couple different explanations. So as Dr. Altieri mentioned, we know that hypoxia can cause bleaching. So it's very possible that the coral host expelled some of its algal symbionts due to that stress of hypoxia, and such that just the overall photosynthetic rate was lower just because they had fewer algal symbionts. We also know that there are damaging molecules that are being created during stress, things like radical oxygen species, other free radicals. So it's possible that these damaging molecules actually damaged either the algal symbionts themselves or their photosynthetic machinery, such that they were just not as efficient at photosynthesis. And that's something that we're still trying to answer. We hope to do an anaerobic metabolite enzyme assay to understand if there were any anaerobic metabolites at work here. But yeah, that's a really great question that we're still trying to answer.
Thank you. One more quick one while we switch slides. I've got one. I'm just wondering if you've thought at all about variation among populations in locations, whether you might expect to get very different numbers in terms of p-crit values.
Absolutely. Yeah. And I think we've heard today that life history has a lot to do with how an organism responds to stress. I would assume that organisms that are periodically experiencing high fluctuations in oxygen will likely do better. For example, in my upcoming studies, some of the corals that we collected actually came from the port of Miami, which is known to be a very stressful, low light environment. So we actually suspect that the corals collected from these more stressful environments will probably have lower p-crits, such that they're able to tolerate hypoxia better. So yeah, life history and genotype will certainly have an effect on the p-crit.
Thank you. Thank you so much.
Thanks.
All right, and now switching from corals to fish. Our next talk is by Lillian McCormick from Scripps on visual impairment as an important sublethal effect of deoxygenation.
Hi, everybody. I am very excited to talk to you. I really wish that I could be there in person. However, I am very excited that I still get to talk to you virtually. So today, thank you for the introduction. Today, I'm going to talk about visual impairment as an important sublethal effect of deoxygenation. So why are we actually interested in examining vision for marine organisms? Vision is very important for a variety of marine organisms for prey capture, predator avoidance. Vision is used for detection of conspecifics for mating or reproductive purposes, as well as light is most often a vertical migration cue for these marine organisms. However, what we know is that vision is highly sensitive to low oxygen. It has very high oxygen requirements, primarily because of the aerobic metabolism that fuels ion transport in visual cells. And the effects of low oxygen on vision in organisms has been described well in terrestrial vertebrates, but not very well in marine organisms. And when we first started exploring this research, we figured out that taxa with fast vision, or animals with high spatial and temporal resolution and very complex eyes, may be most susceptible to visual impairment from low oxygen. And in the marine environment, this is active arthropods, cephalopods, and fish. And most of the work that we've done has been on the invertebrates, so we're going to talk about those today. So I don't need to explain to this audience, globally, there has been about a 2% decline of oxygen content in the last 50 years. So this figure is showing areas with a high degree of change in dissolved oxygen, shown in these red shaded areas. So global deoxygenation is truly a global problem. However, I would like to point out that regionally, the losses of oxygen can be much greater than the global averages. And so, for example, off the west coast of the United States in Southern California, there has been a loss of about 20 to 30% of oxygen on the outer shelf. And this is exacerbated by rising nutrient inputs from coastal systems that decrease the oxygen and cause eutrophication. So not surprisingly, this loss of oxygen across the entire world is going to have a large effect on the animals that reside in these areas. And one of the effects that we've seen of deoxygenation on marine organisms is that it can cause them to change their vertical distribution. And previous studies have shown that even very small changes in light and oxygen, both of which are required for vision, can change the vertical distribution of marine species. And some more recent studies have also shown that climate change can alter the sensory environment for marine animals, whether this is a reduction of sea ice causing a brighter environment for marine animals, or rising surface temperatures causing animals to be distributed deeper in the water, which changes the wavelengths of light that they're exposed to. And within this research, scientists have seen occasionally this disconnect between tolerances observed in the lab versus observed in the field, such as with animal distributions. And the scientists that have observed this have hypothesized that behavior or other sublethal changes may be causing some of this disconnect. So for our research, we were really interested in determining how low oxygen affects vision, and then subsequently the habitat choice in marine animals. And for this research, I focused on early life stages of marine invertebrates in the Southern California Bight, again, off the west coast of the United States. And in this region, oxygen and light, or radiance, vary on diel, seasonal, and interannual scales. So the 4 species we focused on were the California market squid, the 2-spot octopus, the tuna crab, and the graceful rock crab. So 2 species of cephalopods and 2 species of arthropods. And all of these larvae vertically migrate. However, we don't have good high resolution vertical distribution data for these species. And importantly for this study, all of these animals rely on vision for prey capture, predator avoidance, and light is their vertical migration cue. So over the course of several years, we were able to conduct several series of experiments measuring the effects of low oxygen on visual physiology as well as visual behavior in these species. And so for visual physiology, we saw that it's very strongly affected by low oxygen. And so in order to quantify the effects of low oxygen on vision, we came up with 3 visual metrics for oxygen sensitivity, which are V90, which is the partial pressure at which there's 90% visual function, V50 and V10, which is the partial pressures at which there is 50% and 10% visual function respectively. So these figures are showing the mean partial pressures of oxygen at which these animals each have 90%, 50%, and 10% visual function. And so we came up with several conclusions from this study. First is that there are very species-specific differences, which several other people have pointed out today, of oxygen sensitivity. And that even is present when looking at the visual system. And the second thing we wanted to point out is that at very low oxygen conditions, we see a reduction of visual physiology between 60% and 100%. So sometimes this low oxygen renders the animal completely blind. And importantly, a lot of visual change occurs at much higher oxygen levels than the levels that actually blind animals, indicating this is, again, this important sublethal effect. So what we wanted to do was see how this physiological visual impairment was manifested in visual behavior. So we focused on the cephalopods for this study, and we placed these larvae in a tall vertical chamber and measured a change in their vertical position in response to an overhead light stimulus. And so generally before and during a light stimulus, the larvae would be higher in the chamber. And then after the light stimulus was turned off, we'd see this very dependable sinking response of the larvae in the chamber. And we were able to use this to quantify this visual behavior under different oxygen conditions. So here's what some of these data look like. So under normal oxygen conditions shown here on the left, we see on the vertical axis, the zero-centered mean vertical position of the larvae in the chamber. These yellow points are showing the position of the larvae before and during a light stimulus, and the purple dots are showing the vertical position of larvae after the light stimulus was turned off. So again, under normal oxygen conditions, we see this very robust sinking response at all light levels that we tested. So low light levels on the left, high light levels on the right. However, once we started decreasing the oxygen enough, this behavior entirely shut down. And this is showing the squid as an example. Once again, I'd like to point out that a lot of these behavioral changes started occurring at much higher oxygen levels than where the behavior completely ceased, indicating again that this visual impairment can be a very important sublethal effect of deoxygenation. However, laboratory oxygen limits for vision have their limits. And so they're really only useful when you can compare them to the actual environmental conditions the animals are experiencing. And so we came up with the concept of a visual luminoxyscape, or a luminoxyscape, which are combinations of light and oxygen in the environment. And using our laboratory determined oxygen limits for vision, we can then calculate a visual luminoxyscape, which are combinations of light and oxygen in the environment that enable greater than 50% visual function. And so we chose this 50% threshold because it was an oxygen level at which visual physiology and visual behavior was impaired, but it was not quite to the level where these animals are going to be dying. And so we're very lucky in Southern California, where we have this really robust environmental data set collected by the California Cooperative Oceanic Fisheries Investigations, or CalCOFFI. And so CalCOFFI conducts CTD, or collects CTD data as well as biological data off the coast of the west coast of the United States, and they've been doing so for over 70 years, which means that we have this really nice record of both oxygen and irradiance conditions in this region, where we can really examine when and where visual impairment may have occurred for these larvae. And so for this study, we focused on a much smaller subset of sampling sites, and we further split this up into what we called nearshore stations, which are these pink dots, and then offshore stations, which are the blue dots. And this is primarily because the larvae are mostly found in this very, very coastal area. So what this allowed us to do was then examine kind of spatial differences in oxygen and light limitation for vision for each of these species. So in this figure, we see depth on the vertical axis as a function of distance from shore. So near shore is towards the left of the figure, offshore is towards the right of the figure. And this color scale in the back of the plot is showing the average partial pressure of oxygen with distance from shore. And so each of these lines is indicating a oxygen limit or irradiance limit for vision. And so for the green, orange, blue, and purple lines, this is showing the oxygen limit that would cause 50% or greater visual function in these animals. And the yellow line is showing the irradiance limit, where these animals, basically the lowest light that would allow for visual function in these animals. And so what I'd like to point out is that for some of these very sensitive species, we saw that oxygen has the potential to be limiting at shallower depths than irradiance does. And generally, we see that oxygen limitation for vision has more of an effect on visual habitat or potential visual habitat than irradiance does. Similarly, we also saw that for all species, both oxygen and irradiance limited habitat shoals near shore for all of the species, which is very important because this is where all of these larvae live. So then we were able to combine this oxygen limitation and irradiance limitation for vision and calculate what we call the visual luminoxyscape depth. And this is the maximum depth at which animals would experience greater than 50% visual function. And so we used this visual luminoxyscape depth to see how the visual habitat for these larvae changes over time. So we examined data between 1984 and 2019. Each of these black dots is a mean springtime visual luminoxyscape depth value. And we really focused on springtime oxygen levels because that's when oxygen is lowest here off the west coast of the United States. And so we figured that this would be more of an extreme example of habitat compression for the visual habitat. And so we, again, considered this for both the nearshore and what we defined as the offshore habitats for all 4 species. And so for all species, we saw that visual habitat has shoaled fairly extensively since 1984. And this is especially true in the nearshore habitat, where the shoaling has been at a rate of about 1 meters per year, all the way up to almost 4 meters per year for these species. And these trends were all very significant for the nearshore area. So in the offshore, we see a slight undulation in the changes of the visual luminoxyscape depth, which is not surprising because it follows very closely the dynamics or the decadal dynamics of oxygen that we see slightly offshore of the west coast of California. And so to conclude, we see that we have shown that visual impairment is an important sublethal effect of oxygen loss, and there is the potential for visual habitat to be compressed by deoxygenation and coastal oxygen loss. And for the 4 species that we studied, we saw that the visual habitat has shoaled since 1984, as well as some of these larvae species are experiencing oxygen conditions that cause visual impairment right now. And not surprisingly, we would really love to collect some high resolution vertical distribution data for these species, not only to really fully determine the effect of the luminoxyscape on their vertical distribution, but also to determine their true vulnerability to low oxygen for these species. However, the concept of the luminoxyscape and the visual luminoxyscape can demonstrate the importance of examining sublethal effects of deoxygenation on marine organisms. So with that, I would like to thank you and take any questions if there are any.
Thank you. Time for a question?
Nice talk, Lily. More of a comment. It seems to me like this is the opportunity for natural selection to take hold here, because those animals that have more tolerance or better vision at lower oxygen are going to be able to, avoid predators or find prey.
Yeah, thanks, Karen. Great to see you. So I think that's a really good point. And so I'll point out two of the species that we studied, the California market squid and the tuna crab, or Pleuroncodes, the sometimes known as the pelagic red crab. So we saw vastly different oxygen sensitivities in these two species. The market squid was very sensitive to low oxygen, and the tuna crab was fairly robust against low oxygen. And over the last couple of years, we had really warm waters off the coast of California as a result of some larger scale warm blob issues, as well as an El Nino that rolled through. And so we saw actually that tuna crabs started really establishing themselves in these areas, which was kind of a northern extension of their range. And so that was something that we were very concerned about, namely because tuna crabs are a really extreme predator on squid eggs. And so we saw them basically decimating these fields of squid eggs and just vacuuming everything up. And so we were really concerned about the interaction of these two species and how kind of their natural oxygen tolerance may affect kind of the ecosystem dynamics in this area. So that's a really great point.
Thank you so much. And just a reminder that all of the remote speakers can be reached through the Padlet if you have more questions. Our last speaker of the day, and always worth waiting for, is Kenny Rose from University of Maryland Center for Environmental Science, talking about responses of fish and fisheries to hypoxia.
How's that? So I realize I'm between you and the refreshments. So I'm going to try to be a little dramatic and provocative. Unfortunately, I'm also here the rest of the week, so I have to be wary of that. I'm going to tell you that we already saw fantastic science today. We're going to see it the rest of the week, and most of it's pretty useless for fisheries management. And maybe I got some people's attention now. Now I'll go into it. I also want to say Kevin Craig provided all the science that's in here. And all the other stuff is purely me. So don't talk to Kevin. So of course I had to show the Breitberg et al. Figure because it hasn't been shown for at least 10 minutes. I will add, I'm one of the et als, but that doesn't seem to matter. So deoxygenation, it seems so important. And look at this crowd, right? You get fish kills, just visually, it just seems so important. That's the Gulf of Mexico dead zone. So the premise of my talk is, and we heard a great panel discussion about policy and Lisa's work and Gone and Good, all this influencing policy. We're not doing so good on the fisheries management side, especially the strategic and tactical management. But despite my jaded beginning, I will end on an optimistic note. I think that there's a window that we can jump through, and it's not going to be easy, but that we can actually make some progress in the fisheries side. By the way, if you want to get the attention of politicians and the public, impact of fisheries, and I'm speaking commercial recreation, I'm not talking about artisanal, I'm talking about the major managed fisheries, that will get attention very quickly. So what are the reasons why we don't seem to be influencing, in my opinion, fisheries management? Not policy, but management. Because most of our studies, and I'm going to say our, though I never get wet and I never go outside, but most of our studies, we have an unknown subset of individuals from a life stage. And yet most of the fisheries deal with species that have very complex life cycles. And we just know about an unknown fraction of a life stage, what happened to them. So I tell you, 90% of the fish were affected where I sampled. I'm going to ask what percent of that is the population? And that is going to be a fundamental question. And we don't seem to do a lot of that in our studies. So scale is one of them. You have to think of the units of management. The second is vital rates. We heard, and I'm not saying any of these studies are bad, they're great science, but we heard about unhappy fish. We're going to hear about the more confused fish. right? In fisheries, it's mortality, growth, recruitment, movement, and reproduction. And if you don't say any of those, it's a really tough place to make a point and to get some traction. So growth, mortality, reproduction, recruitment, and movement. Those to me are vital rates, right? And so I think we can do a lot more if we work together and bring in, and of course this is self-promoting, bring in modelers and population dynamics people, ignore me, that's what I do, with some of these experimental results and field results, because I could see that you can do this, but we seem to stop before we get to that point. So the third reason we seem to be having difficult traction in the fisheries management arena is we're pretty loose with a couple of terms, and we tend to think that deoxygenation seems to be pretty much an indirect effect, right? And we heard that even from Bill's great analysis. If you notice, by the end of Bill's talk, he was talking about warming, right? Because it's warming through the oxygen effect, right? So there's many types of indirect effects. So when you talk to someone in fisheries about an indirect effect, say what you mean. Does it mean stressor A affects stressor B? Does it mean A and B together cause non-additive effects on a vital rate? Or is it displacement of the organisms and you have indirect food web effects? What do you mean by indirect effects? And as we know, there's many examples, and they're additive, they can be antagonistic, they can be synergistic, and that's all great. But I hear people discussing and the term indirect effects, and I myself don't know what really is going on, because I know a little bit about a lot of things, but I don't know a lot about anything. And so I kind of get lost in those discussions. So given all that, you'd be surprised, I wrote a chapter with others for the IUCN on impacts of deoxygenation on fisheries. And what we discovered when we really tried to dig deep in this was that there are several linkages to fisheries that are very important that we're not taking care of. Probably the biggest effect I can think of that impacts, now remember, I'm not saying fish. Fisheries involves harvest, right? When you say fisheries, is a harvest involved. Otherwise, you're talking about fish. Is displacement and shifts in their spatial distribution. So fisheries has a magic thing they call catchability, Q. And it's, if you walk into a fisheries meeting and you say, I have information that Q is not constant, you will get their attention, right? Because for those who are interested, it's C, catch, equals QF times biomass. Effort is F. That's the whole scaling factor they use in stock assessment. Of course, it's more complicated than that, and there are lots of we talk about all kinds of hyperstability in Q and all that, and there are good examples of hypoxia, probably not killing anything, but changing Q. For example, on the left is a Menhaden study we did, and it's not very clear on here. But what's overlaid there are where the trawlers were dragging, and it's overlaid on the hypoxia map. So they know exactly where the hypoxia is, right? And they know that the Menhaden are going to go in closer to shore when there's a lot of hypoxia, and they plan for it. What that means, though, is that Q is not constant. When there's a lot of hypoxia, you are going to underestimate Q with a constant Q, which means your management advice is riskier than you think. And that's just that's their language. I'm afraid you're going to have to go to their place. They're not going to come to yours. And that's just the way it is in that world. Here's shrimp. This is great. So on the left are where they're shrimping, and on the right's the map of a hypoxia zone. So they're shrimping right along the edge. Why? Because the shrimp come out kind of confused and they aggregate there. So this is what we're working on now, is seeing if this results in a non-constant Q that's related to hypoxia. So that's, I know it's hard, but you can work with people who do speak this language. And it's a strange language, by the way, and I'm in a foreign country. and fisheries is a strange language. They have their home jargon. When I was in fisheries, I was nothing, we were in our own college and our own school. And oceanography was separate, by the way. So we're with forestry and we're with wildlife. Why? Because those are things we harvest, trees, ducks, and fish, right? So it's very different mentality that you have to kind of get used to. And of course, having spent 19 years in Louisiana, I had to show shrimp cooked. You're right, you can't just show the actual organism, right? So there are these shifts that are dramatic that would resonate in a fisheries environment of talking about management. A fourth reason we seem to have trouble, and not policy, but I'm talking about fisheries management, is fisheries management is place-based and it's site-specific. And so it's location, location, location. That's how they manage it, and that's how people deal with it. And so when we have results that are difficult to relate to what we call the unit stock, then it's very hard to make that link. So it's very important that that what you have reflects the nuances of the site where the activity occurs, where the fisheries occur, or where a different life stage is affected at then harvest later. But you really, and I know our job is to be generalist and generalize. And if you want to get into fisheries, you have to be specific because they are managing a geographic area, even though it sounds odd to us. A fifth reason is multiple stressors. And I say that, Denise, because we've known each other over 35 years. It's a mess. Okay, I disagree with whoever was on the panel, but it doesn't matter. Your message gets swamped out. So every time I'm talking in a fisheries group, the oxygenation becomes warming. And somehow, I hope there's no OA people here, ocean acidification is still on the table. And the oxygenation goes away, it becomes warming. And I don't know why. So you have to remember what they're faced with in fisheries. There's all kinds of information. And that's the face, if you didn't notice it, that's like hypoxia and deoxygenation. And it's very hard to get your message heard that way. One way to do this is we did, those who know me, an agent-based population model of winter flounder along the US East Coast. And we did actually, I'm guilty. Look, we did warming in OA, not hypoxia or deoxygenation. But we worked the model so that we reported changes in steepness. So steepness is a measure of stock productivity from the spawner recruit curve. So that's all model generated. And what we showed was that warming alone had a certain steepness. The higher the steepness, the more you can harvest. It's that simple. And when we added OA, there was an interaction effect, but OA alone had very little effect. That got people's attention. Now, they still ignored us, but it got their attention, right? So we had a foot in the door, and then they decided the model wasn't good or whatever the reasons were, something else happened. But so you have to kind of think or talk to people who talk in that language. You walk in, you talk about Q, you talk about steepness, they will resonate with that. They may not believe you, but they'll resonate with that. Finally, the last reason is delivering information. And they're not going to meet us halfway. We have to go into their room all the way. They are so busy and dealing with so many things. And by the way, I've not had a single one of my papers reviewed as closely and rigorously as the stock assessment reports. They redo the analysis as part of the review. So they will check everything. So it's on their terms, how they need it. Sometimes they don't know what they need, right? But they ask for biomass, but they really want productivity. But for the most part, they're pretty savvy about that. It needs to be compelling and well supported. If you go in and say everything's collapsing, they're really not going to listen, to tell you the truth. They're faced with that all the time, right? We often confuse habitat, and we don't define it, with abundance. They're not the same. Habitat, if you ever say the word habitat, or at least when I'm around, say what you mean. What do you mean by habitat? Do you mean growth? Do you mean spawning substrate? What do you mean by habitat? It's a terrible word, because when I say habitat, I bet you there were 10 different versions of what I meant by habitat just in this room alone. And don't confuse it with abundance. It's capacity, right? So, and remember, there's strategic and tactical, and I think we can make a lot of gains on strategic. I don't think we'll ever get the tactical in there. So this was the, and people know this. So the researchers, you listen to our talks, background, the details and the conclusions. You go into, I think policy, this is true too, but you go into a public meeting or the fisheries group, you start with the bottom line, you say, so what, why is it important? And then half the time they don't care about the supporting details, right? So we have to kind of think and package our stuff much better. I like this one myself. The top is saying, and I won't try to repeat, I don't want to go late. I know people are going to get out of here, but it's not only what you know, but they know what they think they want to know, but sometimes they don't know that they want to know it. I'll just leave it at that. The second is, remember that everything is about production, and production is non-linearly related to population size. So you have to know where you are on the population to know if you're going to increase or decrease productivity. Last few slides, I'll go quickly. This is a pet peeve of mine. Fishing is not a stressor. Now, some people are not going to like that. Fishing is an ecosystem service. Overfishing is a stressor. I guarantee you, you walk into a fisheries meeting and you call fisheries a stressor, you might as well go home. You're done, right? It is not a stressor. We do a lot of overfishing, which is a stressor, but don't confuse fishing with stressor. And by the way, in the US and probably ICs as well, it is sustainable to remove a population down to about 20% of its unfished biomass. That's considered good practice. You're removing 80% of its biomass, and they're following the rules of fisheries management. Do I agree with that? Not always, but keep in mind that is the mentality there, right? Okay. The last thing I think is when you're bringing all this, we always bring bad news to the fishers. We never walk into a room and tell them something which increases their catch or quota. We're always taking another slice out of the pie. And so it's okay, because that's probably good management, but keep that in mind that that's their livelihood. And everybody comes in, and because they have the knob you can turn, and you can't turn hypoxia and deoxygenation, you can't turn OA. When you start taking slices out of the pie, they're going to turn down their knob, right? So don't be surprised if you're not welcomed all the time. Finally, there is good news. So everything used to be single species, and we're moving to ecosystem approach to fisheries management, ecosystem-based fisheries management, and EBM. And as we do that, there's much more room for deoxygenation and other stressors to be in there because it's broadening basically the environmental space that they're looking at. So it's very positive that way. And we're progressing. All of a sudden, we're progressing quite quickly up that, which surprised me. It stalled for many years. Finally, the last thing, how can we influence them, right? So advice can be from context to strategic to tactical, right? We fit in with context and strategic pretty well. I'm sorry, I'm pointing to it. I'm not supposed to. And then these are the kinds of things we can do that fit in. Journal special issues, hence us. But also other stuff. There's state of the science reports. There's ecosystem state reports. There's now going to be a whole bunch of FEMs, fisheries ecosystem management reports. It's going to be very hard to get in here. That's a pretty rigid process, but you can have a great influence, still have a great influence in that. So finally, there's a window of opportunity. I've never heard more talk about the oxygenation, and I've been embarrassingly doing this for decades about the oxygenation in fish. And now is a really big opportunity, but we have to climb up that ladder to get there because we have to go to them. And so I'll end with, I know nothing about the subject, but I'm happy to give you my expert opinion. When you go in, remember, these people know a lot about their particular species and fishery. And if they don't know, they hire consultants. So you're not talking to people who are uninformed. And then finally, everything, what does it say? Everything should be made as simple as possible, but not simpler. If it's too simple, it's too general, and it won't be relatable to them. So hopefully, I ended on a positive note, and I believe I'm out of time, and so I will stop with that.
Thanks so much.